Well, I think the very fact that this is the first time that university-related research has been carried on by the chiropractic profession is a great step forward.
The studies going on here have been accepted on their scientific merits, for their scientific validity by the National Institutes of Health.
So we tried to find out first why the chiropractic had such a good result. It's never been documented scientifically. We have to explain what's going on by scientific terminology so that we can bridge the chiropractic profession and the scientific profession.
We know chiropractic works. Now we're about the business of finding out scientifically why it works.
Dr. Sue is one of the nation's leading specialists on spinal biomechanics. He has gathered a team of researchers at the University of Colorado that is making investigations directly related to chiropractic. Research into the biomechanic or mechanical workings of the spine and related studies on nerve effect.
Because chiropractors not only concerned about biomechanics, they are concerning neurophysiology. If we can put together these things, it will be truly chiropractic-related research.
While Dr. Sue's own specialty is spinal biomechanics, he is intensely interested in the work being done in the research paralleling his own. The investigations on spinal nerve roots being carried out by other members of the research team.
There have been studies done on the nerves out at the periphery of the body, for example, the arms and the surface of the body, but none on the nerves as they have exited from the spine. Now, of course, this is where the chiropractor has his interest focused.
Cath, we're stimulating the left sciatic nerve. We're recording from dorsal roots, L5 and 6. We're compressing the root between an inflated balloon about 2 or 3 millimeters in diameter and a plastic foot. The pressure that we apply to the nerve is measured in millimeters of mercury by an electrical transducer. One of the tenets of chiropractic, as I understand, is that compression of spinal roots is responsible for symptoms that can be treated successfully by chiropractors. Now, there's been a lot of work on compression of nerve over the last 100 years, but mostly, in fact, with one exception I know of, all of it has been on peripheral nerve. So we were interested in finding out exactly how sensitive the spinal roots were to compression block. With peripheral nerve, it is virtually impossible to produce a significant block of conduction with less than 150 millimeters of mercury or so. With spinal root, we should be able to get a fairly significant block, at least half of the fibers block, possibly more, with about 30 millimeters of mercury. We're going to start compressing a small part of the nerve. Now, as the pressure increases, you'll see that line, the bottom line on the oscilloscope, the bottom trace, go up.
As the bottom line comes up, the pressure on the nerve is being increased. As the peaks diminish in size, the potential of the nerve to function normally is being reduced.
We just raised to 45 millimeters of mercury. Now, if we wait a while, it should go down even further. It is gradually decreasing in size. This is a storage oscilloscope, and it retains on the face of the oscilloscope the tracing, so that you see a rather dim tracing going all the way to the top of the oscilloscope. That was the original action potential. It's reduced to more than half its peak size. The area is considerably more reduced, in fact. So we're at 45 millimeters of mercury, and we're down to about, I should say, at least 40% of the original value. And we have found, indeed, that the spinal roots are very sensitive to pressure.
We want to know, can a vertebra sufficiently misaligned create a sufficient pressure to change the function of a nerve fiber? And that's what this finding this past year has shown, that you don't need very much pressure at all to change that function.
Everything that we're going to see in this laboratory represents tools in trying to understand how collections of nerve cells operate when they're hooked together. Experimentally, we know a lot about how single cells work, but we don't know much at all about how cells work in networks. Each one of these little boxes represents then an artificial nerve cell, and we try to simulate the behavior of networks of cells by connecting them together. The thing that we're looking at is in reality a tool, and it represents the behavior of a so-called standard nerve cell, even though individual nerve cells deviate from the behavior of this in many different ways. It represents more or less a composite or a behavior of an average cell. It is then a general purpose tool, and we take it then and try to apply it to various specific instances. We use this model to simulate the behavior of reticular formation, for example, a part of the brain stem and various other networks.
And it's the work or the types of investigations being done at university levels by teams such as this that the profession is open to having investigation done that will help draw other students, other people of various interests into the profession to add their contributions, their backgrounds and knowledge.
Dr. Marvin Lutjes has found that nerves subjected to pressure undergo chemical changes.
Well, the approach we're taking here is that subluxation has seemingly resulted in a wide range of physiological dysfunctions or abnormalities. And what we're getting at here is the fact that if you ligate a nerve or compress a nerve, it may begin to start producing toxic substances that can be transmitted throughout a larger area than might have been expected on the basis of either neurophysiological observations or anatomical knowledge.
In this experiment, Dr. Lutjes has used the electrophoresis process to measure the biochemical changes of the nerve after being subjected to large amounts of pressure. The nerve in the experiment is first tied, then extracted from the animal, homogenized, centrifuged, and put into an electrophoretic gel.
An electrophoretic gel is essentially the material that you see here between two glass plates. The proteins that have been obtained from the nerve through the centrifugation migrate through a very gelatinous kind of material like Jell-O, and the Jell-O screens the proteins in terms of their molecular weight, in terms of their size. Essentially, these kinds of studies have allowed us to determine that pressure on nerves, ligation of nerves, does result within a very short period of time, that is, within an hour to 3 hours, in substantial alterations in the structure, the fiber of the nerve, or the molecular constituents of the nerve. In other words, these nerves get sick, they're in a state of ill health, they start to rupture, they send out these toxic materials, and they affect all the nerves in the surround. So it makes for a much more complicated situation than might have been expected otherwise.
Even though I've been practicing for 25 years, if something is demonstrated here that would conflict with an opinion that I've held on to for some clinical reason or otherwise, then naturally, as a person whose chief interest is the welfare of my patients, I would change immediately.
Most important part in this research is to build computer model of the spine. To build a computer model of the spine, we must know the actual mechanical properties of spinal ligament, spinal disc, and other tissues involved in the spine.
The kind of information we want will be more akin to the kinds of repeated loadings that would occur, and this is supposed to be analogous, of course, to action in the human spine, the kind of loading that would occur under normal motions. Now, the function of the dog tendons is to give us some technique in handling the material itself, the tendons. The reason for using the dog tissues instead of using human tissues is that, first, they are much more readily procurable. And they also give us some direct analogies to the testing of human tissues. Certainly, we know that they don't approach the behavior of the tendons in the in vivo condition. But at least we should be able to learn enough about handling the materials in handling the dog materials that when we get into testing the human tissues, we know that we have a basis for comparison. At any rate, the general philosophy behind the testing is to give us data that Dr. Su uses directly in his computer program to simulate the behavior of a real human spine under conditions of normal motion.
Biomechanics research is a new area of the scientific research, and nerve systems study is also. And more importantly, that two things put together have never been attempted before. So this is a completely new research and highly sophisticated research, which require not only that qualified researcher, but require the kind of sophisticated equipment such as computer.
And neurophysiological modeling, biomechanical simulation of the spine, and lots of data analysis, which we do with animals. It's all go through this computing equipment to get data analysis done.
The mathematical computer model of the spine can be visualized by computer graphics.
We have done two-dimensional study because the two-dimensional development will help to develop three-dimensional. The three-dimensional model is our goal. And with three-dimensional model, we can see things more precisely and from many different points of view and prospectively more accurate. It's more realistic. We like to see the scientific improvement of the chiropractic technique itself. If we know why chiropractic is working, then we know how to improve that one more. The chiropractic research must get more attention and find out and why it works and improving this particular techniques, because it has tremendous potential in a future healthcare system.
On Tuesday, December 18, 1973, President Richard M. Nixon signed the Health, Education, and Welfare and Labor Department Appropriation Bill, H.R. 8877, for the fiscal year 1973-74. In the bill, up to $2 million was earmarked for chiropractic research under the National Institute of Neurological Diseases and Stroke. The chiropractic research project at the University of Colorado, initiated by the International Chiropractors Association and carried out by Dr. Su and his research team, played a key role in the proceedings leading to adoption of the chiropractic portion of this bill.